Thank you, Michael, for sharing your thoughts on the situation science is in globally at the moment and how that affects the entire scientific community, but us as ASLO community as well. My name is Patrick Fink from the University of Cologne in Germany, and it's my pleasure to introduce today's plenary speaker to you, who's Dr. Grace Wilkinson. Many of you probably know Grace already very well because she's an integral part of ASLO for many years now, even though she's still, I would say, rather early in her career. But even when she did her PhD at the University of Virginia, she was already an active member of the ASLO community as a student representative. She was part of the ASLO board. She published many papers, including in ASLO journals. She's now an associate editor of ASLO. She's an ASLO fellow, so she's very active in our community, and that's why I guess most of you already know her. And I'm very happy that today she's giving our plenary lecture where she's going to talk about her science. And after completing her PhD at the University of Virginia, Grace was appointed an assistant professor at the Iowa State University, where she, I think, got very much interested in dealing with how the landscape around lakes interacts with processes within these lakes, how nutrient inputs contribute to causing algal blooms, which is part of her research nowadays. And for the last couple of years, she's now, she was first an assistant and then an associate professor at the University of Wisconsin-Madison, where she continues her work on harmful algae, but also is involved in a lot of long-term research. And Michael, just a few minutes ago, emphasized the importance of long-term data and long-term research. She's also involved in whole ecosystem studies, so she has a really broad research portfolio, and many of you have also met her as one of the scientific chairs of last year's ASLO summer meeting in Madison. So it's a great pleasure to welcome Grace here as our plenary speaker today, and I would like to all of you to welcome her with a big applause.
Thank you so much for that. Thank you so much for that very kind introduction, and it's a true honor to be in front of you all today and sharing some of my research. This is a community that means a lot to me, both scientifically and just as a place where I've found so much intellectual growth and community. So thank you. Today I want to talk about resilience thinking for algal blooms. Now, it seems like we're hearing about harmful algal blooms more and more in the news these days. And this is concerning both in the coastal marine environment as well as in lakes and reservoirs, because blooms diminish ecosystem services and can pose a very real potential threat to human health. Blooms threaten things like drinking water, fisheries, recreation and tourism. They can even decrease property values. So of course, it's no wonder that we're hearing about these in the news more and more. These are big events that have really significant impacts. But it also begs the question whether blooms are becoming more common or more severe. And this is a question that I've been thinking a lot about for lakes, which is what I'm going to talk to you about today. And it's certainly a reasonable hypothesis that because large-scale changes in drivers of algal production could be causing widespread intensification of blooms. So, for example, phosphorus pollution is one of those drivers. And while phosphorus pollution is a global problem, many places around the globe, I'm going to focus in just on the United States here and also use this as an opportunity to highlight some of the exceptional science that our federal scientists do. So Sabo and colleagues at the EPA, for example, mapped phosphorus inputs to the terrestrial landscape in the United States. And as you can see here in the darker brown colors, that's where we have even higher phosphorus inputs. So for example, in the upper Midwest, where I'm coming from, lots of phosphorus input to the landscape, and even around the area here in Charlotte, North Carolina, we see a lot. And over 90% of those inputs are from some form of agricultural production. And with all this phosphorus input onto the landscape, not all of it's taken up by plants or removed through harvest. And so there's a lot of surplus phosphorus there that has a high potential for runoff into our surface waters. And speaking of runoff, Precipitation patterns have also been changing, and that's been very well documented by the National Climate Assessment, which is led by the U.S. Global Change Research Program, whose membership is 15 federal agencies. As you can see from this map here, where there's some regions that have been receiving less precipitation, but others, again, for example, in the upper Midwest where I'm coming from and all those phosphorus inputs are, we've been seeing an increase in precipitation over the past decades. And it's not just this gradual change. In fact, what we're seeing is that if we look at just the more extreme events, or those that exceed the 1 in 5 year storm, those have also been increasing over time. So the big storms are getting bigger. And this is certainly changing the timing, the magnitude, and the form of external nutrients coming into our surface waters. Lakes are also warming, right? And in addition to that, it's again, not just the gradual warming that we're seeing, but also the incidence of those extreme events, or heat waves, are changing. And that can have a large effect on algal production. So for example, some recent work coming out of my group led by PhD student Danny Sidlowski has shown that when we have a heat wave in some of the experimental lakes I'll talk a little bit more about here in a few minutes, we see that following that heat wave, there is an abrupt but only temporary increase in chlorophyll concentrations. And it's even more so in those lakes where we've been adding nutrients or manipulating the food web. And so coincident with this algal bloom intensification hypothesis has been growing public awareness of algal blooms. So for example, what you're seeing on this map, if I get it to animate, which I didn't. Oh, I can go back though. Never mind. If you were seeing it animating on this map, you could see that there are points that are showing up for each local news story that has been reported about a harmful algal bloom layering up over the decades. So there's been more and more reporting about blooms in our local media. And so this match of scientific anticipation and public attention, I think, has created the perception, broadly speaking, that blooms are getting worse or intensifying. But human perception of slow change is actually quite imperfect. As my colleague at the Center for Limnology, John Magnuson, put it, some of us remember that there was less snow last winter, or fishing was better a couple of years ago. It's the unusual person who senses with any precision changes occurring over decades. And because we're unable to sense slow change directly, processes acting over decades are hidden in the invisible present. Now, in order to reveal this invisible present, we need long-term data, like Mike was talking about before, the real value of that long-term data, in order to understand the direction and magnitude of long-term change. And that's the same need that we have, need for long-term data to understand if there's been recent widespread intensification of algal biomass in lakes. And so this is a question that I've been thinking a lot about, and with my collaborators, we set out to try to address. And we had definitions for what we meant by recent and widespread and how we were going to track algal biomass. But in order to really answer this question, we needed to first identify what we meant by intensification. And so we came up with 3 metrics. The first is a change in magnitude. So on average, has biomass been increasing from year to year? And if there's an increase in magnitude, that might be tied to an increased likelihood that ecosystem services are being lost. Next, a change in severity. In other words, is the peak of the bloom, the worst part, becoming more and more severe every year? Perhaps in the past, yeah, the lake bloomed, but it kind of turned a green color. Now it's thick, chewy, that pea soup green, you almost think you could walk on it. The lake is so thick with algae. And an increase in severity is likely tied to increasing consequences of that severe bloom. So severe consequences, things like fish kills. And then finally, duration. Is the bloom lasting longer and longer? So in the past, yes, maybe the lake bloomed, it lasted for a few days or a week, but now it's a month or perhaps even the entire summer. And if that's the case, people are going to be less willing to see that as a resource where they can go and recreate or for tourism. And so this could be tied to an increased period of summertime economic loss. So using the incredible database, LagosNE database, that was published by Serrano and colleagues, we were looking for time series of chlorophyll measurements that were taken during the summer, at least every 14 days to capture these 3 bloom metrics. And we found, and we also needed those time series to be for at least 10 years with minimal gaps. And so we found 323 lakes that had time series of chlorophyll A that met these criteria. And we used those in a trend analysis to try to understand how magnitude, severity, and duration was changing of algal biomass across these hundreds of lakes. So what I'm showing you here is the standardized trend coefficient for all 323 of those lakes in our 3 metrics. And if the coefficient is positive, that means we're seeing that metric increase, negative, decreasing. And if we were, based on our hypothesis, we would expect, if there's widespread intensification, that all of these distributions would be heavily skewed towards the increasing side. But of course, that's not what we're seeing. These are very normally distributed. And so based on this population of lakes, we're not finding evidence of recent widespread intensification of algal biomass. And if we dig in a little bit deeper on just those lakes where we saw significant trends, statistically significant trends one way or the other, we found that about 11% of lakes were getting significantly worse or increasing in one of these metrics. But very surprising to us, about 1 in 6 lakes, or 17%, were getting significantly better in one or more of these metrics. And of course, this was a really surprising result. It went contrary to what we were predicting was going to be happening. However, the general conclusion has been corroborated by a number of other studies that you can see listed here. And most importantly, these are studies that are using different methods like remote sensing and looking at different populations of lakes, some of them even tens of thousands of lakes. And even where we see instances in the literature where it seems that we're at odds in terms of our conclusions, so for example, this Ho et al. Study from 2019, looking at 71 globally large lakes, they saw that about 2/3 of those lakes had an increasing trend in severity. Now, the population of lakes in our study was substantially smaller. You can see those distributions of lake size that I'm showing here between our study and the Ho study. But interestingly, what we found in our population of lakes is that those lakes that had the largest surface area in our study were more likely to have an increasing trend in severity. And so if we consider across the continuum of lake surface area across these 2 studies, we might not be in as large a disagreement as initially thought. But this still left us with the question of why so many lakes were getting better. Now, one possibility that we thought about was, well, maybe we're seeing benefits, action taken, restoration, and efforts from the Clean Water Act and other water quality policy. Maybe that's why so many lakes were getting better. It turns out that's a really hard question to answer. There's no sort of centralized database where we can go and find out what sort of restoration or management activities have been taking place in a lake or the surrounding watershed. But what we can do is look to local news and local reporting. So if you're an agency or a lake association group, a watershed association, and you're about to invest a bunch of time and resources into doing restoration, you tend to want people to know about that. Especially maybe if it's after the fact and it's been successful, you've reached your goals, you want people to know about it. So you put out a press release, and those press releases get picked up by local reporting. So we look to newspapers, TV stations, and such for all of the lakes where we were seeing significant improvements. And we found evidence that about in 65% of lakes with significant improvement had some restoration activity that had taken place in the lake or watershed during the period of study. So while that's certainly not conclusive, it is suggestive that perhaps that's part of what's contributing to this trend. What's more, we also found that eutrophic lakes, so those that are already quite green, high in nutrients, they were more likely to have a decreasing trend in both magnitude and severity. And maybe this also aligns or provides additional evidence for this idea, in that those lakes that might already be experiencing harmful algal blooms are already eutrophic, might be where we're trying to put our time and resources into restoring and pulling them back or reducing that eutrophication. But interestingly, this only held, this idea about eutrophic lakes decreasing, if the lake hadn't been experiencing any change in summertime precipitation or even seeing a decline. In lakes like Lake Mendota, which I'm showing here, Lake Mendota is at sort of the heart of Madison, Wisconsin, and the Center for Limnology sits right along its shores where I do my work. Lake Mendota is a eutrophic lake, and we've been experiencing an increasing intensity of summertime precipitation pretty substantially over the past decades. And it's in lakes like Mendota in our data set where we see increasing storm intensity and they're already eutrophic. Those are the lakes that actually have a slight chance of becoming or increasing in their bloom magnitude. And that's definitely what we're seeing in Lake Mendota as well, where we have long-term monitoring to demonstrate that. And so what our analysis revealed, and is supported by a growing body of work, is that algal biomass has not recently been increasing in every lake everywhere. In fact, there's really a diversity of trends in how algal biomass is changing in lakes. And what our analysis sort of revealed is that really this diversity of trends, it's the result of climate interacting with the ecosystem conditions locally, and that's shaping phytoplankton resilience to eutrophication. So we can think about resilience as the capacity to maintain state, so the ecosystem to maintain its state, whether that's in the clear water or low algal biomass state, or the bloom state, the high algal biomass state. And that's done through strong feedbacks that absorb variability, both in internal processes in the ecosystem, as well as external drivers. So for example, in the clear water state, some of those feedbacks, there's going to be nutrients that are coming in from the watershed, that external loading that's going to stimulate algal growth. But we have things like competition for those nutrients by other aquatic primary producers, the macrophytes there, as well as grazers that are helping maintain that lower algal biomass. So in our ecology textbooks, right, resilience is usually illustrated with this sort of cup and ball diagram here. Where the two cups representing the clear water state or the bloom state, sometimes we refer to them as basins, and the ball is the ecosystem or the lake. And currently it's sitting in the in the clear water state or that low algal biomass. And we can think about this entire stability landscape of these two basins, the ball, as well as this unstable threshold or the tipping point at which the ecosystem is going to be drawn by feedbacks into one of the two states. We can think of this as this continuum of algal biomass. So if you went out to the lake, took a sample, and were measuring algal biomass, you would, because the lake is in this clear water state, it would be quite low. But the ball doesn't just stay in one place. Disturbances are constantly coming along and pushing the ecosystem around in this stability landscape. So for example, we might have a storm that comes along. Due to runoff, there's a little bit more nutrient coming into the lake that's going to stimulate algal growth. And if we were to go out and take a measurement a couple of days after that storm, we might measure a slightly higher increase in algal biomass. And so these stochastic disturbances create the variability that we see in our time series of things like algal biomass. But while disturbances push, feedbacks pull, right? And so with that higher algal biomass, that's going to stimulate more grazers and grazer production, which is going to help draw that ball back down into that lower part of the basin at the bottom. And so again, if we were to measure over time, we would see this variability. And because in this particular example, there's such strong feedbacks, the basin walls are quite deep, right? The basin is quite deep, and there's really strong feedbacks capturing the ball and keeping it in that low biomass state. And so we could imagine, say, that this lake with this stability landscape is situated in a watershed that, say, is quite forested. So there's not a lot of nutrients that are cutting off the landscape when these storms are coming by. But say over time, perhaps there's some change in land use, more agricultural production, and that's going to shift the stability landscape. There's now more nutrients that are coming out of the watershed with runoff, and that has essentially changed where that equilibrium of that clear water state is. Now it's sitting in a slightly higher position on that algal biomass continuum, and our threshold is also shifted. Now, when these same disturbances come along and move the ball around in the basin, that storm is washing nutrients, it's going to move even farther away. And so we're going to see that algal biomass is increasing, and there's much more variability within the system. And in fact, those disturbances might even push the ecosystem across the threshold and into that bloom state for a while before another disturbance pulls it back into the clear water state. So when it comes to managing blooms, we tend to think of the stressors that I've listed here and many others individually. But I would argue that they are numerous, they're interacting, and they shape the stability landscape of algal biomass. So instead of focusing on these forces independently, what if we could instead manage for resilience as the emergent property of all the feedbacks, forces, and disturbances that are driving blooms? So a resilience-focused approach to management, for example, would allow us to do things like detect when resilience is eroding in order to use that information to protect humans and ecosystem services as we guide that transformation. Might allow us to use information on mechanisms that confer resilience to spread the risk of major disturbances that are a threat to critical ecosystem services. And I think perhaps most importantly, how can we use that resilience information to try to adaptively manage away from those thresholds and into a safer operating space? But doing any of this requires reliable monitoring, models, and statistical tools. And so it raises the question, do we have the capacity to move resilience out of our textbooks and put it into our toolbox? I would argue that to meet this challenge of making resilience useful for management, whole ecosystem experiments are a really critical tool here. Whole ecosystem experiments can provide evidence for mechanisms, and absolutely, models and smaller scale experiments are critical for building intuition about those mechanisms. But when we want to test that intuition or our knowledge at the scale of ecosystems, because that's the scale of management. Whole ecosystem experiments also allow us to test our capacity to modify resilience, as well as evaluate methods for tracking resilience both over time and between ecosystems. And so over the past decade, I have been a part of and leading teams that have been trying to perform whole lake experiments to understand resilience of phytoplankton to eutrophication. And we've been asking questions like the ones you see here to help us essentially operationalize resilience. And I want to share some of our findings with you here briefly today and our progress. Starting with, are there reliable early warnings before a bloom? So going back to the cup and ball diagrams, if we think about the depth of that basin, let's say we're in an instance like you see over here where we have a pretty steep walls. Those stochastic disturbances, those storms and things are constantly coming along and pushing the ball around in the basin. So if we were measuring the state of the ecosystem or what its position is, say going out and measuring chlorophyll concentrations, we'd see that there's some variability. The ball's getting pushed around, it's moving. But the basin walls are really steep, and so the ball can't move very far. So if we measure variance, it's going to be there, but it's going to be quite low. And if we look at autocorrelation, or how much does today look like yesterday, it's also going to be quite low. Now, as resilience erodes over time and those basin walls become less steep, those disturbances are still coming along, those shocks and fluctuations, and now it's pushing the ball even farther away. And so as we continue to measure things like chlorophyll concentrations, we see that the variance is increasing because the ball's moving farther away and it's taking longer for it to come back. And similarly, we would see autocorrelation increase. And so theoretically, this rise in variance and rise in autocorrelation should be an early warning indicator that resilience is eroding and we might be going towards that bloom state. So to test this theoretical idea, we did a set of whole ecosystem experiments. So we wanted to know whether or not we could pick up these early warning indicators in really messy ecosystems where lots of things are happening. So we did this set of experiments in Peter Paul and Tuesday Lakes. These lakes are located just over the border from Wisconsin in the Upper Peninsula of Michigan. And we added nutrients to Peter and Tuesday Lakes, with Paul serving as a reference. And the idea was to erode resilience over time in Peter and Tuesday Lakes and see if we saw those early warning signals before they tipped over into a bloom. And we were measuring all sorts of things during these experiments and using sensors, like you see that sensor platform pictured there, in order to follow the state of the system. Now, we did this over 3 years, 2013, 14, and 15, and we did the nutrient additions in 2 lakes. So we had 6 lake years of experiments to understand how well these early warning indicators worked. And so what we found across those 6 lake years is that early warning alarms appeared 6 to 57 days before bloom conditions were reached in either of the lakes that we were adding nutrients to. And I think most helpfully, thinking about this as a perhaps predictive tool, is we found that more of these early warning alarms fired off in more variables. So we weren't just measuring chlorophyll, we were measuring phycocyan and dissolved oxygen, pH, all things we expect to change with the state of the system. More alarms in more variables before the most severe blooms, which helps build confidence that when we see a lot of these alarms firing off, we're not perhaps getting false positives, but that is actually heralding a change that might be coming to that bloom state. So this was exciting results for us, but it's still in some ways experimenting under idealized conditions. We know Peter, Paul, and Tuesday Lakes very well. My collaborators, and Mike alluded to this, have been working on these ecosystems for decades. And so when we add nutrients, we kind of know how they're going to respond, and we control those nutrient inputs. So the next step was to figure out, well, how well might these early warning indicators work in the real world, or in ecosystems where we didn't have any of that control, and there was all sorts of stressors and dynamics happening. And so when I was at Iowa State University, a master's student in my group, David Ortiz, who you see pictured here, used sensor data collected in a number of different Iowa lakes. These were lakes that were very eutrophic and were experiencing frequent, large, harmful algal blooms. And he applied these early warning indicator metrics, and he was trying to understand whether or not we can pick up these signals. And what David found was that on average, we saw these early warning indicators, alarms go off 17 days before bloom conditions and 51 days before the peak or the worst part of the bloom. So that even builds some more confidence that at least in these types of systems, we are seeing these early warning indicators fire off. But I'm not here to argue that 6 or 17 or even 51 days is enough time to, say, intervene and potentially pull the ecosystem back from the brink and prevent a bloom in real lakes and ecosystems. And so the way I think about these early warning indicators is how we can use them to be more proactive in our management of blooms as opposed to reactive. So instead of waiting until the bloom appears, going out and testing for, say, cyanotoxins, how can we use this early warning indicators or information to get resources in line, get people aware of what might be coming, protect the public and our health? That's one possibility. So while we were in Iowa, my group also performed another set of experiments trying to test one potentially manageable aspect in lakes, the food web, and how that might alter resilience to nutrient loading. So I've already gone over a bit, but just as a reminder, we know that grazing is a part of the feedbacks that can really buffer phytoplankton from episodic nutrient loading, so nutrient loading that might happen during a storm. And of course, we know from things like trophic cascade theory that when we have an even number of trophic levels, we could expect that there would be more grazing pressure, larger grazer community, and just larger bodied grazers. But pelagic food chains are not the only food chains in lakes, right? We also have, for example, particularly in shallow lakes, these benthic food chains that are another pathway for energy and nutrients and biomass to move through. And these two food chains can be connected by mobile predators like the largemouth bass that you see pictured here, as well as other generalist consumers that make connections between these groups and among trophic levels. And so we can think of both the number of trophic levels as well as the diversity of connections among them as a food web complexity, essentially. And so when storms increase nutrient loading temporarily, so there's this episodic pulse of nutrients coming into a water body, these both pelagic and benthic primary producers are going to be competing for those nutrients, and that energy and nutrients are going to be moving through these many different pathways. And when there's more pathways, it's possible that all of these things could provide stronger feedbacks to help maintain that clear water state. And so we wanted to understand, how does the degree of food web complexity influence phytoplankton resilience to episodic nutrient loading? So to answer this question, in Iowa, we set up these 6 experimental ponds that you see in the picture here. We had 3 food web treatments, so a low complexity, medium complexity, and high complexity with the food web members that you see pictured there. And we wanted to mimic what these nutrient pulses or loading looked like when it came to storms. And so half of the ponds of the 3 food web treatments received pulses of nutrients twice during the summer, whereas the other half served as a reference with the same food web treatment. Now, we wanted to, I said, mimic what came off the landscape in storms in Iowa, and so in general what that was either a 3 to 5% increase in phosphorus, and we usually see a P to N ratio of about N to P, of about 24 to 1. So that's what we added in our pulses. Now, the benefits of this experimental setup was that there were already natural communities within these ponds. We just did the fish stocking in order to create these different food web treatments. And as you can see from the photo, there's effectively no watershed to these ponds. And so we really were able to have confidence that our nutrient inputs, our pulses, were what could be driving some response in the system. And honestly, we set this up to run in the summer of 2020, not anticipating the major changes that were coming for us that spring. And so everybody in the lab was able to pivot and safely do their science in asking questions within these ponds. And so as you can see, we collected just a ton of data. But for the sake of time today, I'm just going to focus on how the phytoplankton responded to these nutrient pulses, which was work that was led by Tyler Butts, a PhD student in my group at the time. And in particular, when I talk about phytoplankton response, we're just thinking about that clear water basin, right? So that blue basin, and thinking about the resistance of the ecosystem, the pond, being pushed out of that low biomass state due to the nutrient pulse, and what the return time to that low biomass state might be. And so Tyler and his colleagues went out every day for 100 days and made measurements of chlorophyll concentrations in these 6 ponds. And what I'm showing you here is a time series of chlorophyll z-scores. This is based on the response detection algorithm that John Walter and colleagues developed. Effectively, how I want you to interpret this is when these z-scores pass the green threshold there, that means there's been a significant response in the pond that was pulsed, so the chlorophyll in the pond that was pulsed, compared to the reference ecosystem and the dynamics happening at that time. And then when the z-scores fall down below this blue threshold that you see there, that means that the pulsed pond has recovered to the reference pond and the dynamics there. And so there's a lot that's happening. You see the 2 pulses being shown by the little rain clouds there and when we added them. I just want to zero in on what happened to the response to the second pulse. And so if we look here to the second pulse, what I'm illustrating is that once that pulse of nutrients was added, the pond that received that had a significant response in chlorophyll above the reference ecosystem within 5 days. So a pretty rapid response. And it took about 24 days to recover back down to the baseline conditions. If we look at the other food web treatments in the medium pond with that same, medium complexity pond with that same pulse, it took much longer for that pond to respond, the chlorophyll to get significantly different from the reference ecosystem. It took about 18 days and recovered within 12, so a much faster recovery as well. And interestingly, in our high complexity pond with both of the nutrient pulses, we didn't see any response different than the reference ecosystem and no recovery because of no response. And so from this experiment, we concluded that greater food web complexity has the possibility to confer more resistance to the phytoplankton, to these pulse disturbances. And so we can infer, for example, from this analysis that there was lower resistance in the low complexity pond and higher resistance in the high complexity. In a more recent experiment that we completed this past summer, back on Peter, Paul, and Tuesday Lakes, we've been testing a new method for tracking how the entire stability landscape, so both basins of the stability landscape, are changed by enhancing resilience through light attenuation. So ecosystems are constantly being perturbed by just a never-ending litany of shocks and fluctuations. And as you can see in this animation here, that is pushing the ball around in those basins, and we can track the position of the ball through, say, measuring phycocyanin, a pigment in cyanobacteria, over time. And so what this animation was doing was building that time series, but in reality, as the observers, we go out to the lake and we have that time series and want to know something about the stability landscape. And so some new methods that were developed by Iranian colleagues a couple of years ago now allow us to essentially take this time series and reconstruct what that stability landscape looks like. And so in doing so, we can also, for example, count the number of times that the ecosystem passes the threshold or crosses it between the two basins. And we can look at how long, on average, the ecosystem spends in either of the basin, right? So we have this distribution essentially of passage times, or how long the ecosystem spends in one basin before passing to the other. And I'm illustrating that here with these green and blue lines. And we can look at this distribution of individual passage times as we've been observing the ecosystem. And in this example, the time that the ecosystem spent in the low biomass basin before passing to the high was about twice as long as it spent in the high biomass or bloom basin before passing to the low. So returning to our previous whole lake experiments, our nutrient additions that I was telling you about a couple of minutes ago, we can look at essentially how the stability landscape changed when we added nutrients to peat. So what I'm showing here is those passage times in a year where we had no nutrient enrichment, no manipulation, we just left peat alone, and a year where we were adding nutrients. And you can see here that with nutrient enrichment, on average, Peter Lake spent more time in the high biomass basin. That makes sense. Resilience was eroded, those shocks and fluctuations were pushing it into that high biomass basin, and it spent more time there. Peter was blooming. But you might recall that we weren't just adding nutrients to Peter Lake, we were also eutrophying Tuesday Lake, or adding nutrients there. And something I haven't told you about the difference between Peter and Tuesday Lakes is that Tuesday Lake is much more stained, and you can see that as an example here. And so in Tuesday, the stained lake, when we were doing those previous experiments, we found that Tuesday required more phosphorus and took a longer time to bloom. And so we hypothesized that light attenuation from the staining provides resilience potentially to eutrophication. And so to test this hypothesis, we added Aquashade, this blue dye, to Peter Lake to make the light attenuation in Peter the same as Tuesday. And I know some of you work in lovely, beautiful turquoise coastal waters, the Mediterranean and things. That's not what is happening here. This is Peter Lake dyed. And I also want to note that the amount of dye that we added to make the light attenuation similar to Tuesday was 0.2 ppm, whereas the manufacturer recommends adding 2 ppm. So we weren't just absolutely dosing this lake with dye, we were adding just a small amount to try to mimic Tuesday Lake. And so then we did the same nutrient loading regime that we had in our prior experiments, and we wanted to compare how passage times changed now that we had increased light attenuation. So in other words, can we purposely enhance resilience of phytoplankton to nutrient enrichment and reliably track those changes in the stability landscape? And I'm showing again here that non-enrichment and enrichment year for comparison, and now we have last year's results of dye plus enrichment. And you can see how the Aquashade not unexpectedly enhanced resilience, and we were able to detect that in these passage times distributions that look a lot like the non-enrichment year. So returning to the fundamental questions to operationalize resilience, we've made some progress. Our experiments demonstrated some reliability of early warning indicators, and there seems to be some promising starts in that we can actually track resilience and changes through time. We also demonstrated how food web complexity might be an important part of maintaining that low algal biomass state in the face of unpredictable disturbance. But this work has also raised new questions. So for example, can we manage resilience without introducing new disservices? And as an example with this Aquashade addition, yes, it increased light attenuation, but that might also be increasing the anoxic volume in the lake, and that will feed back potentially to methane production. So stay tuned for that story. But our next step here is to figure out when and where these strategies or tools might be most useful to implement. As with the early warning indicators, we have a lot of work to understand outside of our experimental systems where these tools have the most efficacy and where the most reliability when we're not the ones pulling the levers on the resilience. So through our analysis of algal biomass in hundreds of lakes, we saw that trends are not getting worse. In fact, they're very diverse. I'm really sorry for that rhyme. But we did see that we know that some lakes in that data set and around the globe are intensifying in their blooms. Other lakes might not be changing in terms of intensifying, but they're already eutrophic, right? And they are experiencing regular blooms. And so what we've shown here is perhaps a promising set of tools for applying resilience thinking to our understanding of management of blooms. We have a set of tools to monitor resilience among aquatic ecosystems, to manage for the risk of losing services. We can monitor and detect eroding resilience to protect humans and ecosystem services. And we have some ways of now monitoring our progress as we try to manage away from bloom thresholds by reducing eutrophication, trying new and creative ways to restore resilience and adaptively manage blooms in the face of a changing world. I want to end by acknowledging my many collaborators for these projects. Without them, this work would absolutely not be possible. It takes a village to do an ecosystem experiment, and you can see there are many villages here. And I'm very thankful to our funders, particularly the National Science Foundation and the Iowa Department of Natural Resources. And with that, we have a few minutes for questions, and I'd be happy to hear your thoughts. Thank you. Your thoughts before you go to lunch.
Hi, Grace.
Hi.
Nice talk.
Thank you.
Oh, whoa, okay. So kind of going way back in your talk to the paper that you presented and thinking about algal bloom trends and things like this. So as you know, I work in Lake Superior. So the situation is obviously a much larger lake, but I think just in lakes generally, we think about them as being more homogeneous than they really are, right? Especially when we're talking about an algal bloom. And so some of the data sets like Lagos are pulling from these monitoring programs where they're taking a chlorophyll sample from the middle of the lake, right? Mm-hmm. And in my experience with algal blooms, both in Lake Superior and in smaller lakes, they tend to be on the shoreline. And so I wonder about like whether we're really representing algal blooms per se, or really just kind of a generalized trend in algal biomass. So you have some lakes, right, where that's extensive, massive algal blooms, and you're capturing that. But I think that there's probably a lot of missed blooms that are in this shoreline area, and we can't really think of them as homogeneous kind of systems.
Yeah, you raise a great point, absolutely. And one of the papers I briefly referenced is sort of in that table, but I think really gets to this, is some work that was led by Ben Kramer, looking at remote sensing of 334 of the largest global lakes. And they really looked pixel by pixel and saw some of those same trends that you're raising, in that if we look at those shorelines, some of those lakes, they are greening more along the shorelines, and it's not as homogeneous. They also showed some really interesting trends, being able to use remote sensing and extend those seasons, that greening might be happening in some of those large lakes more in the shoulder seasons or outside when we might be looking. And so certainly a limitation of our study, relying on data that had been collected in the past by many others, right, in these hundreds of lakes, was the single point location, in addition to having that more limited summertime season. And so definitely that's next avenues in work, and we need to be thinking about those things as well. So thank you for raising that.
Yeah, thanks, Grace.
Thanks for a great talk, Grace. I'm curious, a lot of these excellent long-term and well-studied data sets are obviously coming from a biased set of lakes. How can we harness remote sensing approaches and paleo approaches and things like that to get a more representative look at all these lakes and the trends?
Absolutely. That's one thing we really thought a lot about in our study as well, is that the population of lakes that we are looking at is certainly non-random. If you're investing the time and the resources to go and be monitoring that ecosystem, there's going to be some value to that data set, or reason why. Part of it might be that those are ecosystems that people are interacting with most, or we really care about the ecosystem services there and maintaining them. But as you raised, remote sensing techniques are certainly one possibility. I also think about the possibilities with some of the new hyperspectral missions that are starting to fly, and what we might be able to reveal and understand there and in different drivers. And so we need to be thinking about a lot of those different techniques to get more unbiased samples. And in doing so, again, some of those studies that I referenced did use that remote sensing and are seeing some sort of similar general conclusions as well with US lakes. And so I think we need sort of all of those approaches in order to understand. Thank you so much for that question.
Thank you.
I'm being told that my time is up. I'm so sorry, but I look forward to any further conversation that folks may have. Thank you very much.